So Andrea, do you want me to start exactly at 3 o'clock?
Yeah, let's wait until, okay, we've got people coming in.
Okay, so will somebody send me a chat message when to start?
Yep.
Okay, I've got my chat open, so I should be able to see it.
And we are live on Facebook too, guys.
Good morning. Thank you everyone for joining us today in this program brought to you by the Smithsonian's Conservation Commons and also in partnership with SeaLegacy and Only One. Note that we'll be recording this webinar. I'd like to introduce myself. My name is Nancy Knowlton. I'm a marine biologist. I've studied coral reefs since the mid-1970s. So that's a long time, and as you can imagine, I've seen a lot of changes. I started off as a rather traditional research scientist, but increasingly I have become aware of the importance of taking science and bringing it to policymakers and the general public because of so many changes that are happening in the world's oceans, many of them not good. This has been a long path for me, but it's one I've found very rewarding. And this is a big part of what this whole program is about. You'll hear from a number of Smithsonian scientists who will share their versions of this, this story, and also from Cristina Mittermeier of Only One, who comes to the conservation world from the perspective of photography. And what we're also going to try to do is not just focus on the doom and gloom. Obviously, there are many problems that I mentioned. I've seen coral reefs disappear around the world, for example, but we really want to focus on the things that are improving, what's going right, in the theme of ocean optimism, which was a Twitter campaign that I launched in 2014 and which has since grown into the Earth Optimism program at the Smithsonian. Now, this one of the things that this program reflects, this fact that we're not doing this alone, but rather in partnership with SeaLegacy and Only One, is the fact that no one organization can do this all by themselves. Different organizations bring different strengths. And this is a reflection of, this particular program is a reflection of our now longstanding relationship with SeaLegacy and Earth Optimism. So what I'd like to do to launch this is start with a poll to get us thinking about the way of thinking about science from the perspective of not just scientific papers, but also how you communicate that science to the public at large. So here you see an image and you can vote on which word best describes this image in terms of your personal reaction to it. All right. And here you see the results of that poll. Or rather, I see the results of the poll. You'll see the results of the poll shortly after we finish. Are we going to the next? I'm sorry. Here we go to the next image. Great. Thanks. There's a completely different image. We'd love to see your reactions to it. And I guess you can see the poll results as far as I can determine, but we'll discuss them more in a bit. So I think we're ready for the third image. There we go. 3 very different images. I'm finding this very interesting, seeing how the results are coming in. I think the diversity of results is a nice illustration that a picture elicits very different reactions in different people. Okay, this poll is now closed as well. And there you see the results. Okay, I think we can close these images now. Thanks very much. Close the poll. All right, so looking at these images, I looked at them last night in preparation for this webinar, and it definitely brings to mind the saying that an image is worth 1,000 words. So of course in the poll you were only allowed 1 word, which isn't really fair. It's really 1,000 words. These images can be so evocative, and they can really motivate people to take conservation actions, grab people emotionally and viscerally in terms of their personal reactions to them. And we very much believe at the Smithsonian this combination of science and storytelling, including visual storytelling, is a critical part of all conservation actions. Now, our partners at Only One are master storytellers and visual artists, and through their photos and videos and words, they are painting a picture of ocean conservation, providing a pathway to action with the goal of increasing impact, which is something we all share in terms of a goal. And so for that reason, we're really thrilled to have Cristina Mittermeier joining us here in this webinar. She'll be talking to you for a little bit. And she's the co-founder of SeaLegacy and the co-founder of Only One. She was also the founder of the International League of Conservation Photographers in 2005, which is, in my opinion, the most important marriage of conservation photography that has ever been created. She founded SeaLegacy in 2014, and as a result of SeaLegacy, she has become continued to be involved and grow this program, which is, as a result, she has played a key role in the launch this month of the program Only One. She was a Smithsonian Conservation Photographer of the Year, and she was also named one of National Geographic's Adventurers of the Year in 2018. So I don't want to take any more of her time. So please, Cristina, take it away.
Thank you so much, Nancy. Can everybody hear me? I think so. Yeah. Thanks for that kind introduction, Nancy. I have been such a huge fan of Nancy Knowlton for so many years. She's one of the iconic marine biologists, one of the women that really has blazed a trail for the rest of us in this industry. So thank you for having me, Nancy, and thank you to the Smithsonian. This is an institution that really has found a way to marry science and storytelling to mobilize people. So I'm very honored to anytime be a part of anything that has to do with the Smithsonian. You wanted me first to talk a little bit about the 4 images in the poll. The first one was a sockeye salmon that I photographed in the Adams River here in British Columbia. This used to be a magnificent return of the species of salmon, and over time it's seriously declined. It is tremendously endangered because of a number of issues, but the most important one is just unregulated fishing. This is a fish that has a long life cycle in the ocean. Every 4 years it returns to spawn. And although it has to survive all those obstacles, it is only when it gets here to British Columbia that it's hammered by fishing, and it's one of the worst reasons for its decline. The image of the polar bear I made in Russia. And we are seeing more and more bears that are hungry and starving because they depend so intimately on a stable sea ice surface on which they travel to ambush their prey. So because of climate change, as the sea ice diminishes, polar bears are having a really hard time. The image of the women on the boat was made in Timor-Leste, little tiny country next to Indonesia, on a little island called Arturo Island. Very dry, drought-stricken, difficult to grow any food. So people have taken to the ocean to survive, to feed themselves like they do in so many places. But in Arturo Island, the women have taken out to the ocean as well, and they're free divers. So I had the opportunity to spend time with them in the water. They're very graceful. They call themselves the mermaids of Arturo, and they're really awesome. And I don't remember what the 4th image was, but if I remember during the presentation, I'll say something about it. What I really wanted to talk about today is the journey of how SeaLegacy came about, because it really summarizes the importance of impact communications, of translating science to a way that people can understand it. Paul Nicklin, my partner, and I spent many years as conservationists and photographers working with National Geographic magazine. But we realized that the most important thing we needed in order to tell the stories that we thought were most impactful was to grow our own distribution channels, to tell the stories however we thought was important. So we basically stopped doing assignments for National Geographic in 2014, and we founded SeaLegacy. And in the beginning, the only asset we had was our social media channel. So we started posting. We were posting stories and pictures, and we were putting ourselves in the stories, telling people, how does it feel like to be out there swimming with wild creatures in the ocean? Because one of the things I've realized over many years working in conservation and communication is that when we invite people into the conversation with a storefront that says science, a lot of people who don't have a background to engage with those types of conversations, who do not have an education in science, might feel incompetent and may reject engaging at all just because nobody wants to feel like they don't know. So I realized some time ago that photography is a great way to lower the entry level into the conversation about the environment and about climate change. With a photograph, you can invite people who feel much more comfortable making comments and asking questions. And so SeaLegacy grew very quickly. We realized that people were really reacting to what we were posting online, to the point that pretty soon we collected about 15 million followers among all our social media accounts. But we wanted our followers to be able to do more. I feel like it's very unfair to make people feel concerned and scared if you don't give them an opportunity to have agency. So in about, 2017, we started building the incipient form of a platform that would allow us to turn the likes and the shares of our followers into more actions. And that is a very difficult thing to do. We wanted people to have the opportunity to not only, like a picture, but to be able to do more. So we coded and started building a platform from the ground up. It was this year, because of COVID, that we were finally able to turn all the resources that would have otherwise gone to an expedition into really building this platform. And we don't want SeaLegacy to be the only organization that has the opportunity to use this as their distribution channel. We want this to be a common utility for the entire ocean conservation community. We feel that every conservation group out there needs a distribution channel that allows them to have more followers and more money. And these were the two key things that we were trying to resolve by using photography to engage people. Number one is we need many more eyeballs on issues related to ocean conservation and climate change. The United States, which is the most philanthropic country in the world, Americans donate about $480 billion a year. But of that money, about 30% goes to religion, and then some of it goes to education, arts, health, and only 1.8% goes to a bucket called the environment. It's the smallest part of the pyramid. And so I know that we cannot, resolve the issues of climate change and biodiversity loss by praying. So our goal should be to tip that pyramid on its head and make environmental causes the best funded issue on this planet. I learned yesterday that of all the SDGs, the one relating to life underwater, the one about oceans, is the most underfunded. And yet the ocean is the most important ecosystem on our planet. It really is the defining ecosystem on planet Earth. So with Only One, what we want to do is engage more people and invite people to participate. And I wanted to talk a little bit about the branding anchors for SeaLegacy and for Only One. The most important branding anchor has to be optimism. And last year, there was a beautiful paper that was published in the journal Nature, and it's co-authored by some of my favorite scientists, including Dr. Nancy Knowlton. And the title of the paper is very inspiring. It's called Rebuilding Marine Life. And what it really is, it's a pathway for how do we restore health and abundance to the oceans by 2050. And for me, the thought that we can actually restore health and abundance to the oceans in a generation is just magnificent. So in that paper, these scientists invite us to investigate 6 challenges. They call them wedges that have to do with the issues that we need to resolve in the ocean. And the key thing about these wedges is that we don't have the luxury to tackle just one or another. We have to tackle them all at the same time. The wedges are, we need to protect more spaces in the ocean, create marine protected areas. We need to protect species. We need to stop the flow of pollution into the ocean. We need to restore the habitats that we have damaged, including coral reefs and mangroves. We need to rethink fisheries, the way that we industrially mine the resources of the ocean as if they were infinite. And finally, we need to tackle climate change. And we need to recast the ocean as a solution to climate change, not just as a victim. So this paper is our biggest brand anchor, and we peg everything we do to that paper. And when I talk to Only One in a second, you'll see that the way we've organized it is also through these wedges. We also subscribe to the idea of radical collaboration and to intersectionality, to making sure that every story we tell is inclusive. I'm going to invite my colleague now, Diego Jimenez, if you could share your screen now so that we can share with the audience what Only One looks like. So You can go to only.one and you will land on this page. This is a platform that is a common utility for ocean conservation. It's divided, it's organized into journeys that are pretty much pegged into these wedges. And at launch, we launched last week with great success. We have a lot of people already subscribing and following. We launched with 3 journeys. They're called 30 by 30, talking about marine protected areas. You see, you can log in and you can start exploring. The second journey has to do with recasting the ocean as a solution to climate change. We have called it Green Seas. And the third journey, it's called Material Change. It has to do with addressing plastic pollution. Each journey is populated with a lot of content, beautiful episodic series that you can watch. They're very emotional, they're entertaining. Every one of the journeys has a lot of written articles. Some of them are long, some of them are short, but they're designed so that they're easy to share on social media. They're really designed to feature the work of people on the ground. And the measure of success for Only One is not just that SeaLegacy is one of the founding organizations. We want to have as many organizations as possible feature their own content here. Now, the key to Only One is that every single piece of content provides viewers the opportunity to take action. And because we just launched at this time, we only have 4 actions, but these actions range from being able to sign a petition to being able to donate to a project that actually benefits communities on the ground. Right now we're trying to support a community in Madagascar that's restoring mangroves and actually working on blue carbon credits. And we're trying to support the coral gardeners in Moorea. So it's very inspiring because when people are invited to participate and through storytelling, we show them how we can find a tipping point, engage with beautiful content and storytelling, invite people to help invest and become part of the solution, and then tell you how the story ends, because you're going to be getting reports on how your contributions of money, of time, are actually creating impact on the ground. My favorite thing about Only One is that we're already partnering with over 20 organizations. If we are successful, by 2021, we should have at least 100 conservation groups featuring their stories, featuring their content, channeling followers and funding to their projects on the ground. And so I think that's my time, but with that, I would love to invite all of you to go to only.one and become part of the generation that solves the issues of our oceans. So thank you very much, and I really appreciate being here. And I have to go, but Diego Jimenez from Only One, my colleague, will be staying and he'll be available to answer questions at the end of the session. Thank you so much, Nancy. I'm really honored to be here.
Thank you so much, Cristina. I think your talk perfectly illustrated why we're so excited about the partnership between the Smithsonian and SeaLegacy and Only One. What really struck me was that you took the time and effort as a photographer to figure out what we were trying to say in that paper about rebuilding marine biodiversity. It's not that easy a paper to read. We try to make science papers as accessible as possible, but it really is a testament to the seriousness of making the photography be based in solid science. And so thank you very much, Cristina.
Thank you, I look forward for your brilliant brain and your leadership. And that paper, by the way, if you guys want to read it, it's amazing. It says a lot more. We just try to distill really the essence of it for the public.
Well, the distilling the essence of the public is exactly what scientists need to do more of. And that brings us to our next section of this program. Again, thank you so much, Cristina, which will consist of 6 scientists from the Smithsonian talking about their work. And it will be broken up into 3 sections of 2 scientists each. And after each 2 sections, it will be followed by a Q&A. So please type your questions into the Q&A section of this webinar. And if you could address the question to a speaker, because if we don't get a chance to answer all the questions during the webinar, we will be able to get the question to the speaker if we have it addressed to a speaker, and then we can get the answers back to you. And those questions will be hosted again by Diego Jimenez of Only One. So now it's time to turn, as I said, to the science side of it. And as I say, this is a partnership. And on this side, the scientists, it's very hard, I think, for photographers to work their way through scientific literature. Equally, it's very hard, surprisingly hard sometimes for scientists to, as Cristina said, distill the message down and translate it and communicate it in a way that makes sense if you're not trained in science. And so we hope our 6 scientists do a good job of that for you in talking about their work, which informs and contributes to our ability to make a difference in terms of marine conservation and get the ocean healthier again. So the first 2 talks will be by Mary Hagedorn. She's a senior research scientist at the Smithsonian Conservation Biology Institute. And her talk will be followed by a talk by Davy Kline, who's a staff scientist and coral reef ecologist at the Smithsonian Tropical Research Institute in Panama. So with that, take it away, Mary.
Thank you, Nancy, and thank you, Cristina. That was a very inspiring talk. I really very much enjoyed it. I am Mary Hagedorn from the Smithsonian Conservation Biology Institute and the Hawaii Institute of Marine Biology. And today I'm going to be talking to you about my work on applying human fertility techniques to coral reefs. Now, I know there's probably many of you that are here in the audience that have perhaps friends or relatives that have gone to human fertility clinics to enhance your families. And so you know that this is about freezing cells for 1 day or two days or months or years. And so today what we're going to do is tell you about how we're now applying this to help save coral reefs. As everyone has already mentioned, the oceans are in trouble, and coral reefs are among some of the oldest and most diverse ecosystems on the planet. But in every ocean, they are under siege. We live on an ocean planet where our seas produce more than 50% of our oxygen. And within our ocean, coral reefs are one of the most important ecosystems, as people have already mentioned. Coral reefs provide homes for 1/4 of all marine life on the planet. So they're amazing magnifiers for our planet. And reefs form natural storm barriers for coastlines and cities and homes, protecting more than 200 million people worldwide. I'm certainly one of them. Our house has been protected over the last couple of years, at least 4 times, by the reef here in Hawaii in Kaneohe Bay. At least 500 million people depend on them for their livelihoods, such as food, fishing, and tourism. And finally, reefs contribute over $350 billion annually to the global economy. So reefs are very important. They are important drivers of our economy. They're important for our survival here on the planet. But the state of reefs, as Nancy has already mentioned, is that they are in trouble. And there are things, both local and global stressors that are impacting reefs, such as locally sedimentation, overfishing, and other local aspects. Globally, we are producing greenhouse gases that can both warm our oceans and acidify our oceans, causing stress for our coral that can lead to bleaching, which is what you see here in this picture with this white coral in the bottom right-hand corner of the image, and reproductive loss and potential death for coral. So there's a lot of impacts from bleaching, and bleaching is becoming more frequent across the globe. Many species of coral are now threatened, and as examples, 50% of the Great Barrier Reef has died since 1985. 95% of the reef-building coral in the Caribbean have vanished. Now, as dire as this sounds, there is hope, and I think we do want to focus on hope and optimism in all our talks. So just to give you a little background about coral, coral are animals, and they reproduce. And so here's an example in this blue circle of a coral polyp. and the polyps then divide and it becomes a colony. When they're ready to reproduce, and corals only reproduce for a couple of days of the year, so for about 1 hour each night, they will produce both eggs and sperm. Coral are hermaphrodites. Within this large bundle that you see here, when it breaks up into eggs and sperm, and you can see the white sperm in that video. And so we, as reproductive biologists, we collect those egg sperm bundles, and then we start applying human fertility techniques to those gametes. So today we can freeze and thaw coral sperm successfully, and we have trained people all over the world to do this. In addition, we have also created our own technology. You can see some of it here. We 3D print a lot of our coral freezers, and we send them around the world to people, or we send them our software. But our goal in all of this is to secure as many individuals from as many species of coral as possible. So today we have over 48 species worldwide with many of my partners that we have trained from the Caribbean, Hawaii, French Polynesia, Gulf of Mexico, and the Great Barrier Reef. So what we can do with these banks, with these frozen repositories, is we can create new coral. And here's an example of a coral created with cryopreserved sperm. You can see the 3 little polyps here in its skeleton, and it's growing. And they are developmentally the same as coral that are produced with fresh sperm. But now what we want to be able to do is to be able to use our frozen sperm to help coral by diversifying their populations. And we're doing this thing called assisted gene flow, which is really just a fancy way of saying we're using selective breeding, which has been used by farmers and plant biologists for centuries. And what we'd like to do is create more robust coral by combining genes from other populations. So for example, we have taken sperm that has been frozen for as long as 10 years across the Caribbean, from Florida, Puerto Rico, and Curacao, hundreds of kilometers apart. And we've crossed them with eggs from Curacao. At first, we didn't think it would work because they were so geographically distant, and there was a putative genetic barrier with this population, but it did work. And we have created the largest cryopreserved population in the world, and wildlife population. Humans actually are the largest, but the largest wildlife population in the world using this selective breeding. And these are growing in captivity. They're still in captivity. And we hope at some point they will be able to go out and help diversify the elkhorn coral populations. This is all done with the endangered species Acropora palmata that you see here. And in Florida, the reef track is very depauperate in terms of its genetics. And we hope that in the future that this assisted gene flow population can go out and help those populations. Finally, we are starting a really exciting project that I'm very, very excited about. And we're creating the World Coral Fragment Biorepository, which needs a much better name than it has right now. But our goal is really to save all 1,000 species of coral in our oceans in captivity. And our goal will be to create frozen fragments, coral fragments. Now, as I said, reproduction is faltering with climate change and bleaching. And so we wanted to be able to push our ability to save coral reefs with freezing and cryopreservation. And so we're freezing small pieces of coral about the size ahead of the pin. And these small fragments can add to this bank and help increase the number of individuals within a species that we can save, and also ensure that all the species are saved. So this is just starting, and we're excited that this is going to be happening. We're using the same methods for all the different partners, and for collecting, tagging, genetics, and husbandry. And our hope is that if needed, this could provide an amazing source for reseeding the oceans in the future. So thank you for your attention. We're very excited that you're with us here today.
Thank you, Mary, for that wonderful talk. We're trying to get Davey Kline, our next speaker, up. He's having a bit of Zoom complications, so bear with us for a minute.
While we're waiting for Davey, let me just take the opportunity to remind you to send your questions into the Q&A section of the webinar. We've already got several, and we look forward to getting more. If we can't resolve the technical issues with Davey, then we'll move on to the next speaker and pull back. But let's give it a little bit more time.
Nancy, I'm back on.
Excellent. Hello, Davey.
Hello. Good evening, everyone. Thanks for joining us. So my name is David Kline, and I'm based at the Smithsonian Tropical Research Institute in Panama. And today I'm going to talk about how we can use conservation technologies to save coral reefs. So as Mary mentioned in her talk, coral reefs are beautiful dynamic ecosystems that unfortunately are in trouble all around the world. And the way we have been studying coral reefs for the last 50 years since the advent of scuba diving is to swim along the reef, marking down the species of corals and their health. And it was very slow going, and it was very hard to get information about the health of corals. With the advent of digital photography, low-cost digital photography, and the increasing resolution of photography, we were able to speed this up. And currently, coral reef scientists Can you see my slides now?
You know what, Davey? It seems like about half of us can and half of us can't. Do you want to try to relaunch?
Yeah. Is that working now?
Not for me.
Should I keep going or?
Yeah, why don't we see whether, Molly, do you think you or Sarah could pull it up? And then Davey, I say just keep going and we'll try to catch up to you.
Okay, can anyone see my screen or no?
No, the comments in the chat seem to say no, 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 we can't. Someone at one point could, but yes, a random yes in there. Okay, here we go.
All right, try
Okay,
again.
Sarah, try again.
Can people see that?
I can
Yes,
see him,
We
Davey.
can see it.
It's up
Yeah.
now.
Okay, so I'll keep going. So basically for the last 50 years, we've been studying them basically with a pencil and slate, but we've started to use digital cameras, which has sped things up. And in the last 10 years or so, there's been new imaging technologies like underwater sleds, such as this one used by the Catlin Seaview survey that can actually image kilometers of reef on a single dive. But when you look at the distribution of reefs, which occur in the tropics all around the world, I would argue that we really need to scale up the technologies that we use so that we can study reefs globally and so that we can get the data quickly, much quicker, so that we actually can do things to try to save coral reefs. And I think this is essential so that there will be beautiful, healthy coral reefs by the time my daughter grows up and by the time her children grow up. And in order to do this, we first formed a group within the Smithsonian, including the Smithsonian Tropical Research Institute, Marine Geo, and Healthy Reefs. And you'll hear more about Marine Geo and Healthy Reefs in a couple talks later on. But we also decided to go for radical collaboration and work with groups all around the world that had the best engineers, the best scientists, the best conservation biologists and managers to try to really do something that can make a big difference. So we partnered with groups that include NASA, Conservation X Labs, Conservify, universities from around the world, and we formed this team of really great collaborators and experts who provide very different perspectives on how to save coral reefs. And I'm briefly going to talk about some of these conservation technologies and how we're trying to use them. So to start with, we are collaborating with NASA Ames and Ved Chirias team there. And we're working with satellite remote sensing as well as drone remote sensing to get global surveys of shallow coral reefs. And we're using new technologies developed by this group that will allow remote sensing to see through the water. They call this fluid lensing technologies. And they've also developed custom multispectral cameras that can actually give us some information about the health of the corals. And we're using it on shallow corals, but we're also mounting them on underwater robots so that we can study deeper corals and their health. We're also partnering with Conservify, who are developing custom sensor arrays that we can place on the reef next to the corals. We can also put it on underwater robots that will give us reef scale data on what the key environmental parameters are like. So they measure things like pH, oxygen, temperature, and nutrients, and we can get a really good idea of how these conditions are changing on reefs all around the world. We're also partnering with Conservation X Labs, led by Alex Deegan and Paul Bungie, and they've developed a technology called the DNA barcode scanner. And we are going to mount this system on our underwater robots and use it to detect coral disease outbreaks of coral predators such as crown and thorn starfish. And we're also going to use it to better understand the diversity on coral reefs using a technology called eDNA, so we can find even the organisms that are hiding within the corals. And we're working with teams at the University of Sydney and the University of Haifa to develop these underwater robots that we call AUVs that we're going to mount all these different sensor systems on. And we're also working with a team at Cal Academy of Scientists who study the really deep reefs. They're coral reefs that go down to 200 meters called mesophotic reefs. So we're going to try to study reefs from the surface all the way down to 200 meters and do it in a totally automated way. And eventually we're going to have a swarm of these underwater robots that will be able to get data about key coral reef ecosystems around the world. And we're working with a team of biologists and scientists within the Smithsonian Institution to try to get the most appropriate data for understanding how corals and coral reefs works. And then by understanding the mechanisms for how they deal with environmental change, to be able to use this data for interventions to save coral reefs. But we're generating thousands to millions of images every year, and that's just going to speed up. So we're also using a technology called machine learning, and we've developed a website called CoralNet, where we use machine learning and facial recognition technology, pretty much the same facial recognition technology that's used in platforms such as Facebook to identify the key species of corals and organisms on the coral reefs. And by using machine learning, we can get data from imagery 10,000 times faster and at about 90% accuracy compared to a coral expert. We're also working with a team in Israel to use the newest technologies in 3D printing for printing artificial reefs and also for printing corals. So here on the right, you might not be able to tell, but this is actually a 3D printed coral. And it seems like the fish on the reef can hardly tell that it's not a real coral. So this might be part of the solution for restoring reefs. And these artificial reef structures have been deployed in Israel for over a year. And already there's communities of fish and invertebrates settling on them, which is a really good sign. And we plan to use our system to not only monitor how well reef restoration efforts are working, but also to find ideal locations where reef restoration technologies can be deployed so they can be deployed as efficiently as possible. And then we're also collaborating with some conservation groups, including the Wade Institute and the Healthy Reefs Initiative, to make sure we can best translate our data and make it as useful as possible for local island communities so they can take steps to ensure that they have the healthiest reefs forward moving into the future. And with that, I want to thank you for your attention. I wanted to thank my funding sources. Thank you very much for listening.
So I think we'll now be moving over to Diego Jimenez to help relay the questions that we've received from you. We've actually received 10 questions already. We'll do our best in 5 minutes. So keep your answers, panelists, brief. Thank you.
Hi, everyone. Thank you so much for joining. And yes, I'm part of Only One. I work very closely with Cristina. I'm actually the partnerships and content lead for Only One. So again, just very happy to be here. I'm gonna try to do our best to answer all of your questions. I'm gonna start out with one for Mary. Mary, the question is, how do you make sure the corals you send will be compatible to the areas they're replanted in?
That's a really good question. I mean, certainly with the, excuse me, the assisted gene flow corals, we don't know the answer to that. We know that they have some Florida genes in them. They're half Florida genes and half Curacao genes. And so one of the best ways to test that is to put them into nurseries so that they're somewhat controlled and you see how well they grow. And that would be my first step before putting them on the reef. But you know, in terms of fragments and the sorts of fragments, for example, as part of the biorepository, the Florida Coral Rescue, which is part of our group, has collected 22 species of coral from Florida. And they have over 2,000 individuals. And so they've collected actual mature colonies, which they hope to get back out on the reef in their same locations because that area has been implicated by the stony coral tissue loss disease. So they're hoping to get them back. But I think going forward, if it's a long time in the future, we're just going to have to use nurseries or just slowly put them out.
Wonderful, Mary. Thank you for that. We have a question for David. Are you concerned at all that this assisted adaptation may cause the loss of the original coral phenotypes, or that governments may view this as an acceptable adaptation to unmitigated climate change, giving them the excuse to further stall any actual action to reduce global temperature rise?
That's a great question. I think before we can do anything, we need to do something about our CO2 problem. So that's the #1 issue, but we can't wait for CO2 levels to come down before we do anything to save coral reefs. Basically what we're trying to do is to buy reefs as much time as possible so that by the time CO2 levels have come down, there are still reefs and healthy corals around that can recover and grow to their past levels. So it's super important to not forget about the climate challenge, and that should be our top priority, but we shouldn't twiddle our thumbs while we wait for this global political action that's needed.
Thank you, David. That's absolutely correct. And it's also something that for all of you going into Only One, you might see that some of the actions are supporting conservation projects on the ground. While that's something that we understand people can do, you know, themselves today, of course, the #1 action collectively that we need to do is address our CO2 emissions. That is absolutely without question. Everything else truly doesn't matter if that doesn't change. But that's definitely a great question. We have another one for Mary. From Ariana, she's saying, thank you for the interesting talk. Has there been any evidence of even very little adaptations in reproduction of wild coral in today's warmer and more acidic oceans?
It's a really good question. Thank you for that. I think what we're seeing is that start going in fits and starts. Here in Hawaii, we've had some enormously impacting bleaching events, and that has impacted a lot of the reproduction. But then you'll have several years where it actually looks even better. So we do see, after a bleaching event, we do see the corals recovering, and I can only assume that's adaptations. In Kaneohe Bay, when we had the bleaching event, almost 90% of the coral lost their symbionts. This is the algae that lives inside of them that feeds them. And so, and they completely shifted the symbionts when they got the corals got it back again. Very few of them died, but the symbionts were different physiologically. So we're seeing adaptation, and it's happening. But as Davey said, we want to be prepared for all avenues. You know, we hope that corals stay in the wild. We want them to adapt, but we also want to give them their best shot, you know, in terms of what we can do in terms of science to help them along, whether it's in situ or ex situ.
Great, thank you, Mary. I think we have time for maybe 2 more questions very quickly. There's another one for David. David, what impact do underwater drones have on marine life? How are we sure it's safe?
That's a really good question. So the underwater drones that we're talking about are pretty small. So the largest ones are maybe the size of like a medium-sized dog, and the smallest ones are a little bigger than a loaf of bread. So they're pretty small, and they don't have any exposed propellers or anything like that. So I think there's a lot of design that goes into making sure that they don't harm wildlife. But they also use machine learning for guidance, so they'll actually actively avoid wildlife. And with this, I feel like they've actually had I don't know of any collisions. It might happen every now and again, but they're small enough that it shouldn't do any major harm. So that's a really important consideration, and one that I think the designers and engineers working on the design of underwater robots take into consideration to minimize the impact to wildlife, and also to make sure that the robots keep swimming. So it goes both ways. Yeah.
I think we're going to have to stop now, but I encourage you to keep sending your questions into the Q&A. I noticed some questions coming into chat. If you could re-type them into Q&A, because the chat we're using to make sure our systems don't crash, and the actual content questions we're monitoring in the Q&A section. And I believe that both Davey and Mary will be staying on, so they can, now that they're not talking anymore, can look at the Q&A and start answering directly some of the questions that you've already typed in, since we won't get to all of them. Okay, next on the agenda, we have Matt Ogburn, who's a senior scientist at the Fisheries Conservation Lab at the Smithsonian Environmental Research Center, followed by Emmett Duffy, who is a marine biologist and director of the Tenenbaum Marine Observatories Network. Take it away, Matt.
Thank you, Nancy, and I'm excited to be here and share some of the work that we're doing today. I'm a senior scientist at the Fisheries Conservation Lab at the Smithsonian Environmental Research Center in Edgewater, Maryland. And I want to tell you about some work that we're doing on another kind of reef that is important in marine ecosystems around the world, and those are oyster reefs. Like coral reefs, there are many places in the world where oyster reefs are doing poorly. And so we're launching a research program from our center in Maryland to try to help develop new tools and ways to be able to study those reefs, understand what some of the challenges are, and how successful restoration might be in trying to restore those reefs. As an example of some of the poor condition of reefs are in some places, where I live in Maryland, we only have about 1% of the historical populations of oyster reefs left. So there's a lot of interest because oysters have been a huge part of our culture and fisheries in Maryland for many generations. And there's a lot of interest and effort in restoring those reefs and rebuilding fisheries. We're lucky to also be in Maryland because it's one of the places in the world with the largest oyster restoration projects that are going on. In the Chesapeake Bay, there's a large-scale effort with many partners to restore 10 reef systems in tributaries of Chesapeake Bay by the year 2025. So we're partway into that process. Several of them have been completed and the construction is underway on other ones. And this photo of a barge putting down stone to serve as substrate for oysters to live on is an example of the scale that restoration has to take place to bring oysters back. Baby oysters actually swim up in the water for a period of days to a few weeks and then need a hard place, historically, and another oyster shell to attach to live and grow for the rest of their life. But if there aren't oysters there already, you need to provide some hard substrate for them to live on. So that's what is shown here. We've been trying to apply a number of different tools, like in Davy's talk, to really be able to study the reefs on a broad scale. And that's been one of the biggest challenges, that a lot of traditional ways to study oyster reefs involve either diving or some way sampling oysters from the bottom and bringing them to the surface and measuring them. But that's really time consuming and very cost inefficient. It costs a lot of money. So one of the first things we've been applying, like Davy, is underwater photography. And these are examples of photos from a set of GoPro units that we have mounted on little PVC frames that we lower down to the bottom. And you can see a stark comparison in these two images between a harvested reef on the left from a place called Broad Creek in the Choptank River, which is one of the places that's the most productive parts of the oyster fishery still in Maryland. There are a lot of oysters there, but you notice the reef is pretty flat and has areas of sand like through the middle where oysters were harvested from. The image on the right is from the next tributary over, which is called Harris Creek, which is one of the largest oyster restoration sites in the world. It has about 350 acres of reconstructed reefs that were completed in 2015, and we've been studying what's happened since. And one of our initial studies using these GoPro cameras, we developed an assay that helps us determine what the quality of oyster reef habitat is, from very low quality with very few oysters and very little vertical structure to the reef, to sites that have a lot of oysters and a lot of complexity and other species living there. And what we found is that harvested sites tend to have only about 0 to 3% with high quality habitat. And the restored sites, up to 74% of the sites that we've gone and looked at, have really high quality reef habitat, which demonstrates clearly that at least in the initial years, some of these restoration techniques are working really well. We're hoping to see that continue and continue to use tools like this to study all of the 10 restoration tributaries and then expand out from there to other regions of the world to study how restoration is working. And I wish it was that simple, but unfortunately it's not. One of the things about oyster reefs is that a lot of places they occur are places in the world that are highly productive. There's a lot of nutrients in the water that allow a lot of algae to grow, and that makes the water clarity really low. So it's really hard to see what's in the water. These two images were taken in winter a few years ago when we could see about 10 to 15 feet, which is about as clear as this part of Chesapeake Bay ever gets. But I'm going to show you a series of photos that show you the range of conditions that oyster reefs live in Chesapeake Bay. If you go all the way out to the ocean side, where you have tides that cause the water level to raise and lower a couple times each day, oysters are even exposed at low tide. So this is what's called an intertidal between the high and low tide oyster reef that is near the coast. So we have oysters growing in this condition where they're actually quite easy to study because they come out of the water twice a day. But as you move up the bay and as the water gets less salty, most of the oyster reefs actually live on the bottom of the bay and in water that gets progressively greener and murkier as you move up the bay, to the point where this photo, which is completely black, is an actual photograph from a place called the Chester River where there's so much algae in the water that there's no light at the bottom at about 10 feet. So it makes it really hard to use cameras in that place to study. So we're beginning to apply a variety of different other technologies, things like a sonar, a high frequency sonar that we're deploying here that uses sound instead of light to create images of what's under the water. And that can see through the water that's murky. And so this is one technology that we're using to both see things like the oysters on the bottom and be able to map the oyster reefs using these tools, but also using really high resolution ones like the one shown here to be able to count and measure and study the abundance of species of fish and crabs and other animals living on the reefs. So we're hoping to be able to continue to show the results of restoration to help us learn more about how restoration is working for the oysters and the other species that live attached to the oysters that can be seen in this photograph, like the sponge, that's the kind of light colored species over to the right-hand side of this photo. But also the things like fish, like this striped bass, which is one of our most popular recreational fish species, and blue crabs, the iconic species of Chesapeake Bay, which depend on oyster reefs as habitat as well. So we're hoping that we can study how oyster restoration is benefiting the reefs and the ecosystem as a whole, as well as providing other benefits like supporting other fisheries for different species. So I hope you've learned a little bit about oyster reefs, and I look forward to answering questions.
And now we'll hear from Emmett Duffy.
Hello, everyone. Thank you, Nancy, and thanks to SeaLegacy and Only One for this opportunity to tell you about a subject that I'm passionate about, and I hope you will become so too by the end. We all know that the Serengeti grassland of East Africa is one of the most iconic features of nature and host to abundant wildlife of all kinds. There is also a Serengeti of the sea, an underwater grassland that supports abundant wildlife as they do on land. But this Serengeti is really a chain of underwater grasslands that stretches from Tierra del Fuego in the south all the way up to Iceland and along the coasts of every continent except Antarctica. Recent research shows that there are many wildlife that depend on this resource. 80% of dugongs and manatees spend their lives in seagrass, mostly feeding on it, as do about 60% of sea turtles, and a quarter of dolphins and porpoises spend their lives there, 100 species of sharks and rays. Seagrass meadows are also essential to fisheries, particularly in the developing world. In Indonesia, seagrasses support more than half to almost all of the protein needs of coastal people. And finally, seagrass meadows protect our coastal communities from storm damage and soak up the carbon that our industrial society is exhaling into the atmosphere. But there's a problem. Underwater grasslands and all the wildlife and fisheries that depend on them are in trouble. It's estimated that about 30% of seagrass area around the world has been lost just since we started paying attention to them in the late 19th century. And there's actually a bigger problem, which is that hardly anybody knows about it. So we are working to change that. And I'd like to tell you how. So it's a big challenge. The first challenge is that for most of the world, we don't even know where the seagrass is or what condition it's in. And without that basic knowledge, without science to support conservation, we can't judge its value to wildlife or the vulnerability of fisheries and the people that depend on them or how much carbon it's storing. So this is why we are building the first global map of seagrass using standard intercomparable methods. Much of the world's seagrass is in remote areas, in deep water or in turbid estuaries where it's difficult to see from the surface. So solving this problem of mapping requires a big toolbox and cooperation among diverse experts. So to do that, the Smithsonian has convened over 100 partners from around the world. These are experts in seagrass biology, satellite remote sensing, geochemistry, computer science, and even electronic trackers that employ sea turtles to tell us where the seagrass is as they're moving around and feeding. We are all committed to building a dynamic global map of seagrasses that we can use to track the effects of climate change and other human impacts, and importantly, to identify promising sites for restoration. And so far, we've assembled data from over 700 sites. So one key tool for finding the grasslands and mapping them is satellite sensing, especially in remote areas. So we're very excited to be partnering with the University of Queensland team that leads the Allen Coral Atlas, and who also have imagery that includes seagrass habitats throughout the tropics, which you can see as these dark areas inside of the reef on the left. But we also need to ground truth, or more accurately, water truth, that is, use divers in the water who can verify what the satellites see, help improve the algorithms for remote sensing, and especially explore where they can't see. So the key is bringing the satellite team and the divers together. And then finally, we want these data to be useful and to be used in seagrass protection. And so we're partnering with the UN World Conservation Monitoring Center, who will serve the data and the maps to the organizations who need them to protect seagrass systems. Seagrass meadows are as productive as farmland, and one of the things they do is to store a lot of carbon in their biomass and in the soil beneath them. And one reason an accurate map is critical is that many countries increasingly are interested in using their seagrass assets in their nationally determined contributions to reducing emissions under the Paris Climate Agreement. So seagrasses are assets in fighting climate change. Unfortunately, they are also vulnerable to climate change. Like corals, seagrasses are in hot water. That is, many live near their upper thermal tolerance limits, and heat waves have been damaging seagrass ecosystems in the Mediterranean, Chesapeake Bay, and the Baltic Sea, among other places. So there is some urgency to this task. One of the dangers of a hotter ocean is that it aggravates disease. Eelgrass is the dominant seagrass over much of the Northern Hemisphere. In the 1930s, it was wiped out over much of that range by wasting disease caused by a humble slime mold, which you can see as these brown patches on the seagrass leaves below here. That disease is now back and it's threatening seagrass in various areas. So tracking that disease as it spreads is crucial, but it's labor intensive and requires expert judgment. So we are developing a shortcut around those challenges based on a major field campaign to understand the ecology of eelgrass disease and its sensitivity to climate all along the west coast of North America. One of the really interesting innovations is that we are working with a team of computer scientists and disease ecologists from Cornell University who've developed a cool new artificial intelligence tool that can identify wasting disease with similar accuracy as human experts, but at orders of magnitude faster rates. So this could be a game changer. In addition to that, we are also linking the field sampling on the ground to drone mapping of seagrass color and characteristics at the landscape scale, led by the University of Central Florida. So just in the last few weeks, we've got data suggesting that we may be able to identify disease from the air on the scale of meters within meadows. This is very exciting and could be a breakthrough in our ability to monitor seagrass in near real time. In order to do that, however, we need to scale up the capacity to do that mapping. And so our colleagues at Central Florida have developed a standard training protocol for citizen drone operators that follows Smithsonian Marine Geo's mission to provide free standard protocols that make rigorous science possible for small teams at modest cost. So like Davey, who you heard from, and Melanie, who you will, this is how we bring the world together to achieve global science and conservation. So I'd just like to leave you with two key points. First is that despite the fact that most of us can't see them, seagrasses build vibrant, beautiful ecosystems that provide many valuable benefits for people and wildlife. Economists estimated some years ago that these benefits amount to more than $3 billion a year just for nutrient cycling services. The second point is that the Smithsonian is leading a global coalition doing the science that we need to protect them. So we have the experts and we have the commitment to get this done. We're trying to do the map in an ambitious 3-year plan, and that will be challenging. So we are keen to invite any partners who want to join us in that. With that map and the other tools we're developing, we can manage our fisheries better, protect threatened megafauna better, and quantify the seagrass carbon that countries can use for solving the climate crisis by protecting seagrass. So thanks very much.
Great, thank you so much, Matt and Emmett. We are going to move into questions. We are running a little bit short on time, so we're going to keep it to one question per speaker. The first one is for Matt. Question is, great work, Matt. Do oyster populations spread by themselves in these conditions? If so, how much? We hope they do.
That's a great question. Thanks for that. It's one of the major research questions that we have as well. So in Maryland, there's an area of marine protected areas that covers about 24% of the historical oyster habitat. And the restoration is taking place in a small part of that. Even though it's large scale, it's still a small part of that. So we're studying places that have been restored, places that are in those sanctuaries where people aren't allowed to harvest oysters, but that haven't been restored. And that'll help us to answer your question. I think it's too early. We haven't seen major signs of the restored reefs causing those other places that are protected and not fished, but that weren't restored recover yet. But the hope is that the restored reefs will help repopulate those places, as well as provide offspring that go out of the protected areas and provide additional oysters that can be harvested in the fishery as well.
Wonderful, thank you, Matt. Next question is for Emmett. Emmett, how can AI or other tech be integrated into seagrasses management?
Yeah, there are lots of great opportunities, both at the level of satellite remote sensing and also on the ground. So some of our colleagues have been using increasingly high resolution satellite and drone imagery to try to get at leaf area index, so that the total sort of area of seagrass leaves, which is really important to coastal protection and to habitat. And what we're especially excited about is the possibility of using artificial intelligence to be able to actually see the disease from above, which would be a complete game changer for being able to do that. But also just using that to fine-tune how we can distinguish seagrass from other kinds of plants in the marine environment. So lots of opportunity.
Great, Emmett, thank you so much. Great, we're gonna bring in our other two speakers, our final speakers, that's Steve and Melanie. Nancy, I'm gonna hand it over to you.
Yes, so our two speakers are Steve Canty from the Marine Conservation Program and the Working Land and Seascapes Program of the Conservation Commons, and also Melanie McField, finally, marine scientist and director of the Healthy Reefs Program. So take it away, Steve.
Thank you, Nancy. So I'm going to be talking about mangroves, which have been traditionally considered nothing more than mosquito-ridden swamps. Of little to no importance, and similar to seagrasses, have historically received limited or no management attention. But mangroves are an incredible group of plants. They can form some of the most productive ecosystems on the planet, and support significant terrestrial biodiversity, such as rookeries of magnificent frigate birds, pelicans, cormorants, and boobies, and habitat to a host of marine species, providing refuge and feeding grounds for commercially and ecologically important fish species, such as grouper and snapper throughout different stages of their life cycles. They also support the well-being of coastal communities and have been shown to significantly reduce flood damage to coastal properties and reduce the loss of human life during storm and hurricane events. As we move further into the Anthropocene, an era dominated by human-driven processes, the role of mangroves become more important, not just in adaptation to, but also the mitigation of climate change. However, mangroves are under threat, and it is estimated we have lost between 50 to 85% of the world's mangroves. In the Mesoamerican Reef ecoregion, a marine resource shared by Mexico, Belize, Guatemala, and Honduras, where over 2 million people live in the coastal zone, we estimated the region lost 30% of its mangrove cover between 1990 and 2010. These losses were driven by climate change and direct human activities such as coastal development and agriculture, all of which remain threats. This is an image of Man-of-War Key in Belize, an important bird rookery that has shrunk by 75% in area since 2001. This is not an isolated case. Numerous mangrove keys that serve as bird rookeries are disappearing throughout Belize and other countries. Despite all of these issues, there is cause for optimism. Globally, mangrove deforestation rates are slowing, and in the Mesoamerican Reef ecoregion, action is being taken. In 2019, in collaboration with the Mesoamerican Reef Fund, we co-led a participatory approach to develop a strategy that promotes mangrove management at the regional level. Over 70 people from community groups, non-governmental organizations, government departments, and academia from the 4 countries actively contributed to the strategy. Together, they brought a range of perspectives, expertise, experiences, lessons learned, and a grounding in terms of feasibility for what is possible in each country. The end result has been the publication of a regional strategy for mangrove management, conservation, restoration, and monitoring. With the assistance of a key partner in support and development of the strategy, the Central American Commission for Environment and Development, the aim is for each country to adopt the strategy as a guide for national mangrove management plans, which feed into the regional approach. Whilst an important step, this is just the beginning. We are now using Smithsonian science to support various initiatives to reach the objectives set out in the strategy, such as mangrove restoration. Mangrove restoration projects are increasingly used to bring back mangroves lost, and through the planting of, mangrove restorations are increasingly used to bring back mangroves through the planting of propagules, their seeds or saplings. To support these efforts and improve best practices, we conducted a study that starts at the basic building blocks of life, genetics. Genetic diversity is important as this allows for adaptation to changing environmental conditions and builds resilience within these ecosystems. But how genetically diverse is a mangrove forest? It's actually much greater than we previously thought. In a small area of mature forest, we identified 3 distinct genetic groups, and we were able to detect these differences among the trees at 2 to 8 meter scales. This has significant implications for restoration practices, and as we finalize this research, we will work to integrate our findings into restoration protocols and best practices in the Mesoamerican Reef ecoregion, and also globally through the Global Mangrove Alliance, of which the Smithsonian is an active member. But what do these significant differences in genetic structures at small scales mean for mangroves at the regional scale? Unfortunately, I can't tell you quite yet because we're still working on processing all of the samples and data from the 35 sites. However, preliminary results are showing that there is genetic connectivity between mangroves at different sites located on the same and neighboring islands in Honduras, and also between mangrove island groups in Honduras and Belize. These data suggest that a regional approach to management is a necessity, as genetic or ecological units of mangroves are transboundary, crossing not just municipal, but also international jurisdictions. As we talk about restoration and management activities, there is an elephant in the room: sustainable financing of all of these activities, which is critical for long-term success. There is a potential solution in blue carbon. Blue carbon is where carbon in the form of carbon dioxide is taken from the atmosphere and stored by marine plants in their physical structure, their roots and shoots, and also their sediments. This can be monetized through carbon accreditation initiatives and nationally determined contributions, NDCs, which Emmett already said are part of the Paris Agreement to reduce greenhouse gas emissions. Mangroves can store up to 20 times more carbon than other forests, primarily in their sediments, which makes them an ideal candidate for blue carbon initiatives and an important tool in climate change mitigation. Next year, in collaboration with Pew, WWF, the University of Belize, and Silvestrium, and not forgetting Marine Geo, we'll be conducting a study to provide national estimates for Belize of mangrove and seagrass carbon storage, which will be used in the country's next NDC update. We will also provide training to in-country partners so that future collections and updates can all be done by these partners. Outputs from this work will also be shared with the Belizean Blue Carbon Alliance to highlight the potential of blue carbon of mangrove and seagrass systems throughout the whole of Belize. So whilst there are many threats to mangroves, there are numerous important efforts to reduce the threats, promote their conservation, and to identify sustainable long-term financial options to support these efforts. The role of mangroves in climate change adaptation and mitigation, such as coastal protection, carbon sequestration in the form of blue carbon, providing fisheries habitat and supporting both marine and terrestrial biodiversity is only going to increase over the coming years. Here at Smithsonian, we will continue to collaborate with our partners, old and new, to build and prioritize effective mangrove management, conservation, restoration, and monitoring efforts. Thank you.
Thank you, Steve. Now, Melanie, you're up.
Thank you, Nancy. All right, so I am, you guys seen this? I think it's a little, I am going to be telling you about the power of people and partnerships, mobilizing a conservation army. Get rid of our things. Okay, so this Healthy Reefs for Healthy People initiative really began with an effort to understand and come up with a common vision and common definition of reef health. We started in 2003 with what should have been a fairly straightforward question from one of our main donors, Roger Sant of the Summit Foundation, who wanted to know what's the bottom line on my reef? How do I know if my investments in conservation in the Mar are paying off? And are you actually achieving the goal that we have to improve the health of the reef? So about 3 years later, we came up with this, which is the core elements of the Reef Health Index that we track over the years in what we're calling a report card on the health of the reef. So just like you have to come up with endpoints in a grading system for your kids at school, these are the grades that we give to the reef in the Caribbean in the modern day. And we have 4 main indicators of reef health, and 2 of these are for the benthic and 2 of these are for the fish. And we've come together around this and been able to create what is like a Dow Jones Industrial Average for the reef. So you boil these 4 indicators that are ranked on 1 through 5 down to a simple number. And so this can be tracked over time. For the last 12 years, we've tracked this. We were doing good. We improved from a poor condition to a fair. And then just in this last report, it actually dropped back down to poor. And so that enables us to look a little more closely into it. That's the 10,000 foot version that I just showed you in the Reef Health Index, but we actually have these data much more refined at the site level. You can track it by country. You can look at the percent of reefs that are in different conditions. And then you can look at the number of surveyors and the number of organizations that are contributing to this. So in this last survey, we had 26 organizations and 82 surveyors. Looking just at for one of the countries, so we can look at MPAs, group together sites from one MPA, or all the sites in one country. We wanted to look for the impact that one of the first recommendations we had made in the report card in 2008 was to protect parrotfish. Belize did it in 2009, and we could see in this graph that, you know, it took 10 years, but we have finally had a increase in the parrotfish and a decrease in the macroalgae. We can go into these data on a live tracker that's on our website, on the healthyreefs.org website, and you can drill down into specific sites. You can look at countries. You can look at the actual numeric values for the coral or the fish biomass that are there. You can look at the depth and the date of the survey. This was for one of the very good sites that were on the west coast of Glover's Reef, which is inside a marine reserve. So these things are all very useful for the practitioners, the scientists in the region, and the managers that are managing these marine parks. And they are really the core of this army for conservation. This is who it's like boots on the ground, where these are the fins in the water, and these are their tools, and they're pretty straightforward, but they require a lot of training and continual training. We've trained over 250 biologists in the region and gotten some that are actually up to trainers so they can actually train more people. So this is a continual process. And this is one of the ways that I think we've been able to solidify this Healthy Reefs initiative and make the partnership valuable to everyone, because it actually is providing a service for all of these partner groups. Sometimes it's not the data and the information, the science that you need to capture, though. It's the hearts of the people. And I think looking at imagery, and we've talked a lot about photography and the role that that plays, you know, sometimes a picture is worth 1,000 words. And this one was called Coffee with Sharks, and it was helping to get public support for the creation of a shark protected area in Roatan in Honduras. And it was successful. And now we're trying to build support for tracking and understanding this disease progression. Mary mentioned it in her talk. It's called stony coral tissue loss disease. We want people to be so interested and worried about this disease that they will actually submit data, and also just people that are tracking it, all of the businesses that rely on the reef, the diving industry. They're very concerned about what's happening with this disease as it progresses through the Caribbean. It was in Florida for a number of years. For 4 years, it didn't seem to move out of the state. And now it's throughout 15 different countries in the wider Caribbean, including both Mexico and Belize in the Mar. So how do you create this, this energy and getting the public and the awareness and raising the awareness? It's a constant interaction that we have with the Healthy Reefs team. Is 4 country coordinators and a communications specialist. And they are constantly attending workshops and working on local issues or national issues that are going on in their countries and dealing with the media. And I think whether it's on an international scale or a local scale, you've got to be listening to what's going on, and you've got to be responsive to what the needs are in your community. And that's how we're reaching a much wider and more diverse audience. So just to kind of summarize how we pull all these different elements together, we start with the science. That's the database, the monitoring, getting everybody trained and using the similar methodologies. With that, we can create these report cards that have recommendations for management actions. And that's where you actually change things and can do something to help the reef. We also need to track and evaluate if those policies are being improved, and are the recommendations you're making, are they actually being implemented? That's what the eco audits do. And we've done 3 of those so far, and we've had 6 report cards over the last 14 years. Within all of this, the only way that it gets power or traction is through the media. And again, launch events and frequent and wise use of the media. So and then it cycles back into more monitoring of the health of the reef to see if things are actually improving as you do all of these things that you say you need to do. And you have to continually evaluate if it's working or not. So the big royal we in all of this, the little Healthy Reefs logo, is all of these groups. And that's what makes it happen. We need all of them to be able to get this work done. But these are the converted, and we need to preach to more than the converted. And I think, this is how we get back to that people power idea, is how are we going to engage with a much wider audience? We need to energize and mobilize people in the way that like these world leaders have on different issues, on different decades. We've got to figure out how to get the ocean science and the needs of the communities and the reefs into the stage of this kind of mobilization. I think when people do come together, you can do amazing things. We did this in Belize in 2010, getting people to coordinate and for an aerial art event that went to the COP climate change convention that was in Cancun. And that's where I'll stop and just say thank you. And again, recognize all of the key partners in the Healthy Reefs Initiative.
So Diego, a few questions?
Yes, thank you both, Steve and Melanie. That was great. Yes, the first question is for Steve. If tomorrow all the mangroves disappear, what is the carbon impact?
It would be huge. So if they were to disappear, it's not just that the carbon itself goes, but the land goes. And a really good example of this is if you look at Guyana and Suriname, which are losing up to 500 meters of coastline a year because of the mangroves disappearing there. So yes, there's a lot of carbon going out. So that would obviously have impacts to climate change, but there's a bigger thing that you have a lot of land erosion and you lose all of this habitat. So carbon loss is horrendous, but the biodiversity and real habitat loss are probably even worse. And the fact that coastal communities could actually get washed away, I think is more of an immediate worry.
Thank you, Steve. Yes, hopefully that is not possible. And that's why we're doing all of this work among so many organizations to prevent that from happening, and also get people to understand that, you know, as you said, mangroves are not just swampy, mosquito-ridden ecosystems. They're actually incredible life-sustaining ecosystems. Last question for Melanie. How do you incorporate the culture as it relates to resource use and management when working with diverse stakeholders?
Well, yeah, that's a very good question. And it's essential to keeping communities engaged, because we don't generally take the front seat in the driving of all of this. You know, the science is there supporting the background, and we're always beside our local partners. So many of these areas have a local NGO that has community members that are actually running the parks. They're the stakeholders. That's who we're training to do the reef conservation, and we generally let them lead. They make all those decisions as when is the right time, how do you push, what do you do. We're just there beside them to make sure that they understand the science and to make sure that they've got the support they need in whatever way that is, getting international attention if they want it, and if they don't, if they say it's not the right time, then we listen and we back off. So I think it's being respectful and treating them as an equal partner in the whole system.
Great, Melanie, thank you so much. Great answer, very true. Nancy, I'm gonna hand it back over to you.
Okay,
Thank
we're
you.
at mine. Can you see? Yeah, let's see. I think I should be on the screen. We're at minus 1 minute, so I'm going to make my comments very brief. Maybe I think I need to have you stop sharing the screen so you can see me, but I'll just keep talking. So first of all, I want to start by thank you with thank yous. I'd like to thank the scientists who did such a good job at sharing what they've learned in a way that was really accessible. And also they've done a really good job at answering your questions. They've answered, there are 6 open questions, and they will still be online answering more questions, but they've managed to answer 27 of them already during the course of this event. And so thank you also to all of you for participating. More than 200 people were online at this webinar, and we really appreciate your thoughts and your comments and questions. I felt that this was an event that really captured the essence of the Smithsonian, which is, of course, an amalgamation of the arts and the humanities and the social sciences and the sciences, and it's both a serious research organization and a major platform for sharing and communicating with the general public and policymakers. And that holistic approach is really a big part of what the Smithsonian is all about. It's also an organization that is both local and global, and really believes in the link between science and storytelling action and impact. And of course, these are all aspects of SeaLegacy and Only One as well, which is why this partnership works so well. Partnerships really are the key. And we are all connected in this. I like to say that we are all citizens of the sea. And because the solutions come from so really any possible sector, as I say, from the sciences and arts, education, communication, technology, we all have a role to play. So I really want to thank you again. On the screen, I think you see the contact information for how to reach us. If you have comments, suggestions, of course, we like to hear how we could do better, but we also like to hear what we like to hear that what we did well, but we also like to hear what we could do better. And as I say, the scientists will be online for a while, still answering a few more open questions, one now, but keep them coming for a little bit. All right, thank you so much. It's been a pleasure hosting this, and I look forward to doing it again.